Well, thank you for having me. I'm really excited to talk to everyone about what I'm passionate about, which is algae and most recently Didymo. Um, Trying to make it so that I can see the rest of my screen and get rid of let's see, there we go. Everyone's on the side now. Yeah, so anyhow, my background is in algae. That was what my dissertation work focused on down in Ohio at Kent State University. I've been with Michigan TU for about 2 years now. Been working on a number of different things from dam removals to in-stream habitat work to invasive species, and specifically Didymo. So today I'm going to talk a little bit about Didymo. Of course, now my slides aren't progressing. Um, there we go. Okay. So just a little overview. So I'll talk a little bit about what Trout Unlimited is, what we do, what invasive species are, and why we care. Considering this is a series of invasive species talks, I won't linger on that for too long. I'll talk about algae and diatoms and why we care about Didymo, some potential consequences of Didymo in the state, and then what we're doing to better understand it and how you can help if you're so interested. So, Trout Unlimited is a nonprofit that extends throughout the US. Here in Michigan, we're focused on, I guess really across the country, but specifically here in Michigan, we're focused on conserving and restoring our cold water systems. So that usually entails removing barriers, things like dams, fixing faulty road stream crossings, anything that inhibit aquatic organism passage. We also are doing a lot of work to improve habitat. So a lot of that is deploying temp loggers and understanding where there actually is cold water and then doing some habitat mapping, looking at the type of substrate, what's around for aquatic organisms to live in, and trying to make that the best that we can. We try to take a sort of watershed-wide perspective when we're doing work across the state so we can strategize appropriately. And then we do a lot of education and outreach, trying to teach people about cold water conservation, various aspects of it, and trying to get people involved with it. And part of that is thinking about aquatic invasive species. And then not per se me, but my boss, Brian Burrows, does a lot of advocacy and policy reform work. So a lot of what we do, we try to be as data driven as possible. So from like I said, trout perspective, we know that trout like cold flowing water, diverse habitat for all life stages, and plenty of food. So based off of that, we know what we can measure to try to better understand what they need. So we, again, look at things like temperature, the habitat characteristics, macroinvertebrate community, as well as invasive species presence or absence. And all of that goes into how we inform our management and make management decisions. So with regard to invasive species, just knowing who is present, where they're present, how they're distributed is really critical, and that's something that's not very well understood with Didymo, as I'll get into that here soon. So just a little bit more on invasive species. Um, Most of you have probably heard of the term invasive species before and probably know that they're species that come from somewhere else. They're usually moved around by people. Sometimes they have other avenues of introduction, but usually it's people. Um, why do we care about invasives? Well, they can have a huge impact on our native species and our ecosystems. You can think of an invasive as an organism that grew up somewhere else, is really good at kind of being a generalist. So they're good at moving around, traveling, and taking over where they land. So, um, with respect to Didymo, I'll get into this a little bit more later on, but we're thinking of Didymo as an invasive, although it's kind of questionable in the state. It's definitely native to the Northern Hemisphere, but um, right now we're kind of at the point where we know it's in a few places and we're trying to limit its spread. The issue with invasives is that once they establish and really form a strong population stronghold, they're really difficult to eradicate and remove. So for many invasives across the state, we're to the point where we're trying to control and just maintain our native species as best we can, and that becomes very costly. The best way to manage invasive species is to limit their spread in the first place. So with Didymo, that's one aspect of what we're trying to do is to limit their spread and to inhibit them from making it to new rivers. And then another topic that'll come up, what about problematic native species? So sometimes conditions change or a new strain of a species is introduced potentially, or a mutation occurs, where a species becomes problematic. Maybe that species has been around and doing its thing and wasn't a problem, and conditions change, and then it starts to take over an ecosystem, grows uncontrollably, and becomes a problem. And that might be what we're looking at with Didymo. There's some evidence of that as well, but just wanted to put that term out there. A nuisance species is a species that, is a nuisance in some way, shape, or form that, doesn't necessarily have to be an invasive. It could be a native species. So invasive species management can take a lot of different paths. I just wanted to touch on some of the common invasives across the state. So if you've been um, paying attention to invasive species work, you're probably familiar with many of these. They span everything from fish to plants, to tiny microbes like Didymo that can grow to be more than just a microbe. So it can be visible to naked eye, but they can take a lot of different forms. Oftentimes they're accidentally introduced with Didymo. Didymo can be transported on wading gear, so like waders or boots, boats, and it can actually survive out of water for many days. Some say up to 40 days out of water, especially when it's grown to large biomasses like what's seen in this picture. So it's very easy to spread it around and think that you're doing the right thing by maybe letting your gear just dry, but sometimes that's not good enough with these. So we'll talk more about that too, about what we can do to best protect from spreading invasive species around. And as I mentioned in the last slide, we're concerned about invasive species because generally they reduce the biodiversity of ecosystems by kind of outcompeting native species. They can alter habitat and really just throw entire ecosystems off kilter. And they generally stress native species extensively. So, and here's just some more invasive species across Michigan. The one that I'll talk about a little bit is New Zealand mud snails, as they seem to appear in similar places as Didymo is appearing. But just wanted to give you this list so that you're aware of some of the other invasives across the state to watch out for. Okay, so now I'm going to transition to talking specifically more about Didymo and starting off as just discussing what algae are, because I think algae are kind of a mystery to a lot of people, not something that most people are thinking about. Algae is a very catch-all term. It encompasses a lot of different organisms. So those organisms are usually photosynthetic, and they're usually living in water. However, there's many algae that actually live on snow. They can live on like damp surfaces on the land, so they're not always limited to water bodies, but usually we think of them in water. They're often microbial, which means you can't necessarily see them with the naked eye, although many can grow to biomass levels where you can see them with your naked eye. Sometimes they're single-celled, kind of free-floating, living on their own. Other times they're colonial, which can be in the form of sort of like globs, which is what Didymo often grows into, or filaments. And overall, algae are very, very diverse. So they encompass everything from cyanobacteria, which are prokaryotes, like bacteria, to a variety of different groups of eukaryotic organisms. And the group of organisms that Didymo belongs to is the diatoms, which I'll touch on next. But the main thing to know about algae is that they're essential for our aquatic ecosystems. So they're the base of the food chain. They are the ones that are not creating, but transforming carbon, CO2 from the air into a form that can then be eaten by everything else in the ecosystem. They're growing, like in this picture in the bottom, they're growing on the surface of rocks, on the macrophytes or plants that are growing in the water. They're free floating. They're kind of, all over the place in most aquatic ecosystems. And they produce much of the oxygen we breathe. I actually recently read a paper that used to be something around between 20 and 50% of the world's oxygen was thought to be from algae. Now they think maybe somewhere up to 70% of the oxygen we breathe is actually from algae. So, they have a huge impact on the global cycles as well, as being important in the individual ecosystems that we're studying. So they're very necessary. They can also be problematic. So, maybe if you pay attention to environmental news, you've maybe heard of harmful algal blooms, which are the instance where, we do things on the land, such as agriculture. We add a bunch of nutrients to our ecosystems. They make their way in, and essentially we over fertilize the algae that are growing in our downstream ecosystems, like our lakes. So like the western basin of Lake Erie, Green Bay, Saginaw Bay, most of the bays around the Great Lakes have harmful algal bloom issues, which are caused by excessive nutrients. However, Didymo is the one instance of an algae issue that is actually driven by low nutrients rather than high nutrients, and we'll talk about that more here soon. They can also cause just nuisance growth. So one that people often complain about is Cladophora. It's a filamentous green algae that grows and covers the bottoms of lakes and rivers. It looks like mermaid hair is what it's been described as, long green filaments. And generally it grows when there's too much phosphorus around. It just outcompetes other algae and grows like crazy and produces a lot of biomass that is less than ideal for many reasons. But in general, you have to remember all these critters are very critical to our ecosystem functioning. So oftentimes we demonize algae because they cause problems that we don't want to deal with. But oftentimes we are the reason why those problems exist. So, uh, okay, I won't harp on that any longer, but just know that algae are essential. Okay, and then diatoms, like I mentioned, are a form of algae. So they're unique in that their cell walls are actually made out of glass. So that's, nothing else really does that. They're pretty unique in that feature. Their cell walls are often very ornate and shaped. Their morphology is indicative of who they are, although some diatoms look very similar but yet are different species. So um, now that we have genetic tools, we have a better ability to understand how all of these organisms are related to one another. They're a common component of biofilms. So a biofilm is, if you ever walked around a stream and slipped on a rock, it's likely because there's a biofilm growing in there that contained a lot of diatoms. So if you look at the surface of pretty much anything in an aquatic ecosystem, it's going to have a layer of organisms, mostly composed of algae, but can also include fungi, archaea, other bacteria, all sorts of things. Diatoms are unique in that they usually thrive in cold water habitats and high light environments. So they're often the most dominant organism in biofilms like this time of year, up until about the leaves start coming back on the trees when there's lots of additional light. In the streams and the water is still really cold, that's when diatoms are usually dominant. However, they're pretty much around year round at some level of relative abundance in our biofilms and our streams and lakes. Diatoms are especially good fish food and invertebrate food. So most algae, when they photosynthesize, they store their energy in the form of starches or like other carbohydrates. Um, Diatoms are unique in that they store a lot of their energy in the form of lipids. They're actually the base of like those omega fatty acids and lipids in food chains. So when you think about fish having, like being a source of fish oil or fatty acids that people take supplements of, a lot of that actually stems from diatoms and it's accumulated and used in fish. So diatoms are really important for food webs. They also have a lot of uses. So because of their unique cell structure being glass, when they die, they don't decompose very quickly. They actually sort of fossilize in the sediment. So you can go out and take a core from a lake and you can see what diet, you can clean the core, get rid of everything that's organic and just look at the diatoms that are present. And you can see how conditions of the lake change based off of what diatoms are present through time. As different diatoms grow under different conditions. And that science is known as paleolimnology. Diatoms are also used in a number of applications in industry, such as in filters. Diatomaceous earth is a common additive to filters. So lots of uses for humans. Didymo, or rock snot, as it's so lovingly named, in appearance looks like this. So it's a variety of diatom that looks a lot like a champagne bottle at the cellular level. It has this a unique ability to form stalks. So instead of just laying flat on the surface of a biofilm in a river, just kind of hanging out on the top of a rock, maybe when conditions aren't right, it can grow these stalks and put itself up into the water column and kind of flap around in the water column rather than laying down flat on the rock. So um, kind of a unique ecology in that aspect. When it grows to biomass levels that are visible to naked eye, it's because it's forming those stalks and forming them very quickly. And actually, when the stalks form and the cells divide, the stalks also divide. So it just grows pretty much exponentially when it's under favorable conditions. This picture is actually from the Manistee River back in 2021. So Didymo blooms when it, again, when it forms these biomass levels that are, visible with the naked eye, are sometimes mistaken for combined sewage overflows. So, or, an issue, a septic or sewer issue, because sometimes it can look like toilet paper stuck on the bottom of rivers. It kind of has this grayish appearance once it's dying. Diatoms in general, as you can tell by the color from the cell picture, often are golden brown. They're actually referred to as golden brown algae. So when Didymo forms blooms, often it's not that bright green color that we associate with other algae. It's more goldeny brown, sometimes olive green, but more goldeny brown color. And then as the cells maybe die or start to decompose, it oftentimes turns this grayish whitish color. And thankfully, where we've seen it in Michigan, Didymo hasn't overtaken rivers as it has been noted to in other places. So, what we see here in this picture, it's kind of wispy. It's covering part of the substrate, but it's not fully engulfing the bottom of the stream, which is an important thing to consider when we are managing for it in the state. So, from a Michigan standpoint, Didymo has been confirmed on 4 rivers. The Boardman, the Au the Manistee, and the St. Mary's. We only found cells of it on the Au last year when we started looking. Prior to starting to collect samples and look for it, you know, the state didn't have a ton of resources dedicated to studying Didymo, so they had taken maybe 30, 40 samples over the last handful of years looking for Didymo in different places, or, if they had received a call for suspicious algae growth, they would go out and take a sample. But otherwise, there hasn't really been um, an effort to take samples and look for Didymo across the state. Because it's a microbe, because you can't see it with your naked eye unless it's growing to these big biomass levels. We wouldn't know it's there unless we're going out and scrubbing rocks and looking at some cells under the microscope. So, it's likely that it's in more places than this. We just don't know yet. And that's part of the project that I'll talk about here soon, trying to better understand the distribution across the state. So, Didymo as an invasive in Michigan. We know that Didymo is native to the northern hemisphere. It's definitely native out west. It's thought to have evolved as a Rocky Mountain stream diatom. So think crystal clear water, snow melt fed, really low nutrient, really high light. That's kind of its habitat preference where it thrives at. It's definitely invasive to places like New Zealand, likely Patagonia and Chile as well. New Zealand, the New Zealand strain is actually thought to be from Alaska. So people can take samples from all over the world and kind of study the genetics and figure out where different populations stem from. The New Zealand strain is from Alaska, and that strain is thought of as being more prolific and growing to denser biomass levels than some of the other strains, uh, as far as I can tell from the papers I've read on it. Stalk formation is what makes Didymo a nuisance species. So if it's just a single cell growing in a biofilm, it's not it's not a problem. It's just there with the rest of the diatoms and algae in that biofilm until it starts forming stalks and really growing to these biomass levels where we can see it with the naked eye. That's when it becomes a problem. So, just want to remind everyone again that diatoms aren't a problem unless they start to grow to nuisance levels. Otherwise, they're critical for our rivers. So we shouldn't just demonize all of them. And some more information about, Didymo's ecology. So, again, we seem to consistently see it blooming under these four conditions. However, there are some exceptions, and I'll talk about that a little bit here soon. But generally, it's that low bioavailable phosphorus, so the exact opposite of harmful algal blooms. For Didymo to be a nuisance or a problem, it only occurs under low phosphorus conditions, thought of to be like 5 micrograms per liter or lower. High light conditions. So in order to photosynthesize and have enough carbon to form those stalks, it needs a lot of light. So it generally doesn't seem to grow under low light conditions. I have some anecdotal evidence just from talking to anglers and fishing guides that when it bloomed on the Manistee a few years ago, they would see it blooming in the middle of the river in places where it wasn't shaded. And then you'd get to a bridge and it'd be like a distinct line where it just stopped growing and wasn't under the bridge where it was, perpetually shaded. So pretty interesting that that seems to be a controlling factor and definitely a good thing to know when we start thinking about how to manage for it. Another thing that Didymo seems to need is consistent flow. So it's thought of as mostly being an issue in rivers and streams, not so much an issue in lakes, especially inland lakes. I haven't heard of it growing in any inland lakes to at least to nuisance levels. It might be there just from being washed downriver from places where it's at in rivers, but doesn't seem to take off in lakes. However, there is a population that grows on the north shore of Lake Superior. And the idea with that is that something with the wave action, the low nutrients, and the light that's available there. It seems to thrive right on part of the North Shore of Lake Superior. However, I haven't heard of it being documented anywhere on the South Shore on the Michigan side of the lake. And again, that cold water. So just a diatom thing. Diatoms can grow in warm water conditions, but usually they're outcompeted by other algae, like green algae, cyanobacteria, which are known to prefer and thrive under warm water conditions. Diatoms are usually more dominant in cold water conditions. Okay, so digging a little bit into the nutrient situation, there have been a handful of studies outside of Michigan. So this is one from Connecticut, where they looked at a number of different rivers. So just number of rivers on the y-axis, and then the x-axis is concentration of soluble reactive phosphorus or bioavailable phosphorus. So really low phosphorus conditions, they saw Didymo forming stalks or just present in, let's see, forming stalks in 4 rivers and present in 2 more rivers. At kind of moderate, still low concentrations, but moderately low between 5 and 10. Didymo was present, but it wasn't forming stalks or causing visible blooms. And then at higher concentrations of bioavailable phosphorus, it's thought that other algae outcompete or that Didymo just isn't triggered to bloom, and it actually wasn't present or found in any of the streams surveyed at that concentration. Um, Again, that's in Connecticut. Here in Michigan, we know that Didymo was present on the Upper Manistee, and at that time there were some water chemistry samples collected, and it seems like phosphorus was like hovering right around 10 at that time. So it might be slightly different here in Michigan than what's going on in Connecticut. So all the more need to study it more here. And then just thinking about some of the bottom-up effects. So this is one of the more popular pictures of Didymo. This is from New Zealand, a river on the South Island that is totally covered in Didymo. So this is, thinking about the consequences of this level of growth. You can just imagine, it's smothering all of the habitat, the benthic habitat, clogging up that habitat so that your macroinvertebrate community is impacted. There's also consequences of mat decomposition. So this much biomass, once it starts to decompose, when it's sunny and the sun's out, Didymo is still going to be photosynthesizing, producing a ton of oxygen at night. All those cells are going to transition into respiring, just like you and I. So they're going to take in oxygen and breathe out CO2. And then when this all starts to decompose and bacteria, take over and decompose all this, they're going to use up a lot of oxygen to break down all this biomass, which also uses, it can deplete the oxygen in the water. So it's thought of as, there's a lot of kind of effects of this level of biomass, but one is, reducing oxygen for other organisms to use, and clogging habitat. And then just some effects on the macroinvertebrate community. So there have not been very many studies on this, but it does seem that when you have a large biomass event, like what I just showed you, you see a reduction in your EPT taxa or your sensitive taxa, so your Ephemeroptera, Trichoptera, Plecoptera, your caddisflies, mayflies, and stoneflies that are, thought of as being like the sensitive taxa that are present only in good conditions tend to go away, or their abundance decreases when Didymo blooms are present. There is some planned studies this year where there's going to be more work on this in Michigan, which is exciting, so that we'll hopefully be able to gain some insight as to how the macroinvertebrate communities here are impacted by Didymo. It does seem that under just kind of low biomass events, so what we've seen on the Manistee and Boardman, where it's not necessarily covering the entire river, it's just kind of patchy, that probably isn't going to have a detrimental effect on these, EPT taxa populations, although it might still reduce the diversity and abundance of those organisms. So we'll know more in the next few years about that. And the consequence, so when these guys go away, often they're replaced with more tolerant taxa. So you think of things like midges, snails, things that are just more capable of living under variable, low oxygen, lower quality habitat conditions. Okay, so what are we doing to, better understand and, try to figure out what's going on with this Didymo issue? So generally, TU tries to be as proactive as possible. So we are trying to monitor the spread across the state. One way we're doing that is through a citizen-led monitoring effort. So we've made a bunch of sampling kits. We've distributed about 1,700 kits to different conservation districts, TU chapters, just any group that's interested in sampling or any individual that's interested in sampling. We've mailed kits out to, and folks are able to go out to a river, regardless of if they think Didymo's present or not. You can go out and scrub some rocks, collect a sample, and send it to me, and then I can look to see if Didymo's present. And that sampling effort is accompanied by a survey that allows us to timestamp and to geotag the samples where they're collected and to gather some information about the streams, just so we can try to start getting at where Didymo is present or absent across the state and what conditions are like, where it is and where it isn't. So we did receive some funding. We're working with Eagle and the DNR on this project. And this project also includes a water chemistry component to better try to understand that threshold of phosphorus that Didymo seems to start forming those stalks and become problematic at, as well as just creating a network of folks who are interested in Didymo and are working on it, trying to better understand it. It seems like there are a number of people that have been working on Didymo. So there's a group at Oakland University who's been looking at decontamination methods for New Zealand mud snails and Didymo. They've found that 409 solution is, just your household spray bottle 409 solution that you can get at most stores. If you douse your gear in that, you should be good. It's effective at killing Didymo and New Zealand mud snails, and it doesn't seem to have a deleterious effect on gear. So that's, they're a group working on that. There's a group at LSSU under Dr. Ashley Murky who's been working on better understanding the ecology of Didymo. So they're right on the St. Mary's River where Didymo seems to be pretty common up there. And they're trying to better understand its ecology with some flume experiments, doing different nutrient additions and different, just putting Didymo in different conditions and seeing what happens, basically. So bringing all these people together, trying to, get people to talk and share their findings and work together rather than kind of doing their own thing across the region is part of this project's goal as well. And here's our website. I can put this up later on too, or just send it out in a follow-up, but you can scan this with your phone to be taken to our Didymo website if you want more information and how to get kits and, take these samples if you're interested. So, some of the key questions, like I mentioned, we are, trying to better understand that concentration of phosphorus for Didymo stock formation in Michigan rivers. So, to do that, we're collecting water samples alongside benthic algal scrapings at 3 rivers, and I'll show you that in a second. Kyla's actually very involved with that on the Boardman River. She's collecting weekly water chemistry samples and diatom or benthic algae scrapings so that we can see if Didymo is present, and when it starts forming stalks, what the conditions, the water chemistry conditions are like over the next 2 years. We're also thinking about some other environmental variables. So I mentioned light and phosphorus and temperature. We're also looking at conductivity, the substrate type. So is it sandy versus gravelly? And some flow data for those rivers to better understand, why Didymo pops up in certain places but not in others. And then, like I mentioned, we really don't have very many samples across the state. We're starting to accumulate those now through this program, but we're just encouraging folks to participate. If you're out and about, you're going to be on a river, get a sample kit and download the survey on your phone to be able to take while you're out there and take some samples. It's, a relatively easy and not too time-consuming process, and it helps us to better understand where Didymo is at across the state. The one thing that's hard with trying to understand the distribution of microbes essentially is that they're really small, and Didymo might be present in a river only at certain times of the year. It might only be in certain parts of the river. So the more samples we can get all over the place at all times of the year, the better chance we'll have at understanding the distribution across the state and gaining some insight into Didymo's ecology and kind of what makes it tick. So just some results from that presence absence sampling so far. All of the green dots indicate Didymo was not found in the sample. The red dots indicate Didymo was found in the sample. So far, we have about 100 samples. Like I mentioned, we've distributed about 1,700 kits. Our goal is to get at least 1,000 of those back so that we'll have samples to look at and populate this map with. This map is available on our website, and I update it as soon as I look at samples. So folks who submit me samples can go and look at this map and see what the sample, status is. And then, when I find Didymo, I let folks know that I find it as well. But again, we're hoping to fill in this map. Right now, it's been pretty, sampling has been localized to certain regions. We're hoping to sample across the state and really better understand where it's at to help us, one, understand, is Didymo just present across the state and the conditions aren't right, so it's not becoming a nuisance or, growing those stocks and, becoming a problem, or is it actually only in a few of the rivers across the state and, that tells us that maybe it actually is truly an invasive. It's been introduced to those regions, and we really need to step up our education, outreach, engagement to try to limit it to those rivers and not allow it to spread to new places. And again, so this is that weekly water chemistry sampling. So here's our locations for where that's taking place on the Asable, the Manistee, and the Boardman. So every week, Kyla and a few other folks are out there taking a water chemistry sample, scraping some rocks, and over the next 2 years, we'll have data that hopefully will provide some insight into when Didymo blooms, what the conditions are like. Worst case scenario, or maybe best case scenario, is that we don't have any more Didymo blooms in the next few years, and that we wouldn't maybe gain as much insight as to what what the conditions are like when Didymo blooms, but we at least will gain insight into what the conditions are like when Didymo is not blooming. So regardless of if it pops up or not, we'll learn something about it, but we will learn more if it does pop up again in these rivers. So. And then what you can do, so if you're interested in, sending me samples, the Benzie Conservation District does have a bunch of kits, and they're sort of leading the charge up in your area. Let's see, Traverse City Conservation District. I don't know if that's the exact name of that group, but they also have kits. And then I can also send more kits to folks if you're interested. But the main thing really is just spread the word, talk to people about invasive species. A lot of people are clueless and have no idea that invasive species are a problem. Or they don't think that their activity has anything to do with invasive species. But anyone who's going out, spending any amount of time in rivers, even if you're walking around, if you're floating in a kayak or whatever you're doing out there, if you don't clean your gear or clean what comes into contact with the river, you could potentially be spreading around invasive species without even knowing it. So, talking to people, letting people know that one way to, decontaminate gear is 409 solution. Just spray your gear, douse everything down. They say to let it sit for at least 10 minutes, 20 minutes is preferred before you rinse it off. I will be honest, I oftentimes don't have a chance to rinse it off. I'm just spraying and going. With field season, being out and about, it can be hectic and it's not always easy to rinse gear off. But I think to myself that you know, not spreading invasives is more important than maybe introducing a very small amount of 409 solution to rivers, even though, that's not ideal necessarily either. Um, I think one of the best ways is to, you can freeze your gear for at least 5 hours. That should take care of both Didymo and New Zealand mud snails and make them both not viable to reproduce. So that's, if you're hesitant to use any sort of chemical on your gear, and you have a chest freezer, toss everything in a garbage bag, throw it in there for a little bit, and that should do the trick too. So with that, I'll take any questions anyone might have. If you're interested in, learning more, let me know. Happy to send along more information. And
Thank you.
here's my email if you wanted to reach out to.
Thank you so much, Jordyn. That was incredible. I'm going to invite anyone to throw questions in the chat or feel free to unmute, turn your camera on, whatever you'd like to ask your questions to Jordyn.